All right, today we are diving into one of biology's greatest detective stories. We're going to crack the code on how a single, simple cell gets the secret instructions it needs to build an entire, incredibly complex organism.
Really think about that for a second. Every single one of us, you and me, we all started out as just one fertilized egg. So how on earth does that one cell give rise to everything else? The cells that let you see these words, the cells that are pumping your blood right now, the cells that are thinking, they all came from that exact same starting point. How do they know what to do?
Well, the answer lies in this beautiful process called differentiation. This is basically the biological magic where these uncommitted, kind of jack-of-all-trades cells go on a journey to become highly specialized experts. They form every single distinct part of a body. You can think of it as the ultimate career path decision, but for a cell.
So let's actually follow that journey. How does a cell go from having, limitless potential to having a very specific, totally locked-in destiny?
It actually happens in stages. At first, a cell is what we call unspecified. It's like a brand new graduate. It could become absolutely anything. Then it gets a little more focused. It becomes specified. Now it has a career path in mind. Maybe it's thinking, I'm going to be a nerve cell. But, and this is key, if it gets a better offer or, different instructions, it can still change its mind. And finally, it becomes determined. This is it. This is the point of no return. It's locked into its fate, and it will become a nerve cell no matter what.
Okay, so that's the theory. But how do we actually know when a cell has crossed that line from flexible to stubborn? Well, scientists figured this out with some incredibly clever, almost elegant, transplant experiments. Let's take a look at how they put a cell's commitment to the test.
So first things first, you need a baseline, a control. This is called an orthotopic graft. You take a cell from an embryo that's supposed to become, say, part of the head, and you just put it right back in the same spot. And, no surprise, it develops exactly as expected, into part of the head. This tells us what should happen under normal circumstances. But now, watch this. This is where it gets interesting. We do what's called a heterotopic graft. We take that same future head cell, but early in its development, and we move it to a totally different neighborhood, the future belly region. And what happens? It doesn't form a weird, misplaced piece of head. No, it completely changes its mind and becomes a normal part of the belly. This is proof. It shows the cell was only specified, not yet determined. It was listening to its new neighbors.
But, and this is the crucial experiment, if we just wait a little longer and then do the exact same transplant with a slightly older cell, boom. The cell moved from the head to the belly, completely ignores its new surroundings, and develops into head structures anyway. It was already determined. It had passed that point of no return and was sticking to its original plan, no matter what. So this tells us that cells can be either flexible or totally single-minded. But it just opens up an even bigger question, right? How do they get these instructions in the first place? Well, it turns out there are two main playbooks that they can follow.
On one hand, you've got something called autonomous specification. You can think of this like the cell having its own internal pre-written blueprint. On the other hand, there's conditional specification, where a cell gets its instructions by listening to signals from all its neighbors. It's really like nature versus nurture, but on a cellular scale.
So let's dig into that internal blueprint first. How does a cell follow a script that it just inherits?
This is autonomous specification. And the idea is actually pretty simple, but it's incredibly powerful. The parent cell will place specific instruction molecules, we call them cytoplasmic determinants, into just one part of its goopy interior, the cytoplasm. Then, when that cell divides, only one of the two new daughter cells gets these molecules, and that inheritance alone seals its fate. It doesn't matter what its neighbors are saying.
And the proof for this, it is just wild. Scientists were able to literally scoop out that little patch of cytoplasm with the determinants inside and transplant it to a completely different part of another embryo. And the result? The organism grew a whole second ectopic head. Come on. It's this brilliant, dramatic proof that these molecules act like a master command switch.
But, not all cells are these rugged individualists just following an internal script. A lot of them are actually getting their instructions by listening to the crowd around them. And this is our second playbook, conditional specification. For these cells, their fate depends entirely on their position and what signals they're getting from their neighbors. If you move the cell, it's just going to become whatever its new neighbors tell it to become. For these guys, the old real estate mantra is absolutely true. Location, location, location. So, how do cells talk to each other? Well, they use these chemical signals called morphogens. Imagine one cell, the source, is just pumping out a morphogen. This chemical then spreads out, creating what's called a concentration gradient. So there's a lot of it close to the source, and then less and less the farther away you get. Other cells can actually read this gradient like a cellular GPS to figure out exactly where they are and what they're supposed to become.
The classic analogy for this is called the French flag model, and it's perfect. Just picture a line of cells. A really high concentration of the morphogen signal tells the cells, okay, you become blue. A medium concentration tells them, you become white, and a low concentration tells them, you become red. And just like that, one single chemical signal creates 3 completely distinct bands of different cell types. It's so elegant, and just incredibly efficient.
Now, the theory is really elegant. But how do scientists actually prove that a specific chemical is really acting as a morphogen in a real living thing? It's time for a classic case study where they really had to put on their detective hats to crack the code.
And our detective story is going to star the zebrafish embryo. These tiny, transparent embryos are a total favorite for developmental biologists because you can literally watch development happen live, right under the microscope. They are the perfect little organisms for figuring out these fundamental rules of life.
So scientists had a hunch that a morphogen called nodal was patterning the embryo. To test it, they did a direct experiment. They injected different amounts of the nodal signal into these embryos. And the results were just striking. A high dose turned on one set of genes, which we see here in red. A medium dose activated a totally different set in green. And a low dose, that turned on a third set in purple. The cell's response was clearly completely dependent on the concentration.
Now, this looks like a smoking gun, right? But any good scientist is always going to be skeptical. I mean, could something else be going on here? Maybe nodal just kicks off some other chain reaction of signals, and it's not actually the thing doing the long-distance work. How could they be absolutely sure?
This is where the final definitive proof comes in. They transplanted cells into the embryo that had a broken nodal receptor. You can think of it like their cellular radio was busted. So these cells were physically sitting right there in the nodal gradient, but they couldn't hear the signal. And guess what? They didn't respond at all. This was the final clue. This proved that nodal itself was the long-range morphogen that was directly telling the cells what to do. Case closed.
So let's just quickly recap the secret instructions that we've managed to uncover. We've seen that a cell's commitment to a job can be temporary, that's called specification, before it becomes permanent or determined. And we've seen that they find their path using one of 2 major strategies, either by following an internal script they inherit, these cytoplasmic determinants, or by listening for signals, these inducing factors from their neighbors to find out what they're supposed to be.
And all of this leaves us with one final and pretty tantalizing thought. For centuries, we've just been trying to decipher the instruction manual of life. But now that we're actually starting to understand the rules that cells follow, the next question becomes, can we learn to write the instructions ourselves? To guide cells toward repairing our bodies, to regenerating what's been lost? That's the future this science is building.